Lab 6, Passive Transport, Part E, The Diffusion of Solutes Across a Permeable Membrane, by Emma and Micah. Let's start off by talking about diffusion. Solute diffusion occurs across a semi-permeable membrane along a concentration gradient from regions of high concentration to low concentration. And a concentration gradient is the difference of concentrations of solutes between two regions, as we can see in the picture to the right of the screen. And the second law of thermodynamics states that the transfer of energy from one form to another increases entropy, or the degree of disorder of a system. And entropy is also known as the measure of randomness of molecules in a system. Because of this, diffusion is a spontaneous reaction because there is an increased amount of free energy due to the highly ordered molecules. Free energy is also used to promote change or do work. Therefore, free energy causes molecules to move from a high concentration to a low concentration. Chemical equilibrium is when the rate of formation of products equals the rate of formation of reactants. And when equilibrium is reached, no concentration gradient is left. And as we can see in the diagram below, we start off with the solute all on one side of the membrane, and over time, the solute diffuses through the semipermeable membrane, and thus it reaches equilibrium where the solute is in equal concentrations on both sides. For this experiment, we recreated a cell and its extracellular environment. Cell membranes are selectively permeable to solutes based on size, charge, and polarity. Using a dialysis tube, we recreated the cell membrane. And the dialysis tube membrane was permeable based on size. First, we filled our artificial cell, which is the dialysis tube, with a solution to recreate the cytoplasm of the cell. Then we placed the artificial cell into a beaker of solution that represents the extracellular fluid. And our ultimate goal for this experiment was to determine the direction of solute movement based on size and the presence of a concentration gradient. For this experiment, our hypothesis was that the smaller the molecules of the solute, the more likely they are to diffuse through the membrane. In this case, the membrane would be the dialysis tube. So for our first part of the procedure, we set up our artificial cell and the extracellular environment. And these are the steps that we used. So first we located the 25 centimeter length of dialysis tubing and folded over and closed off one end with a dialysis clip. Then we placed the opened end of the dialysis bag over the stem of a clean funnel and filled it with 25 milliliters of the starch Na2CO4 solution. Then we folded the open end of the dialysis bag over, squeezed from the tied end to remove as much air as possible, and closed that end with a second dialysis clip. Then we rinsed the bag off in the pan of deionized water and gently patted it dry with a paper towel. Lastly, we submerged the dialysis bag into its beaker solution, which was also the extracellular fluid, and we recorded our starting time, and we allowed the dialysis tube to remain in the beaker for 60 minutes. Our second part of the procedure consisted of us determining the direction of solute movement. So first we used the marker to label test tubes 1 through 8, and after the 60 minutes was up, we poured 20 milliliters of the cytoplasm, which was the liquid within the dialysis tube, into the 25 milliliter graduated cylinder that we had. Then we poured 5 milliliters of this solution into test tubes 1 through 4. So test tubes 1 through 4 each got 5 milliliters. Then we cleaned and dried the graduated cylinder that we used and poured 20 milliliters of the extracellular fluid, which was in the beaker, into the graduated cylinder, and poured 5 milliliters each of this solution into test tubes 5 through 8. Our last part of the procedure consisted of us testing for direct solute movement. So firstly, we would perform the starch test on test tubes 1 and 5. So for this, we would add several drops of Lugo solution to each test tube, and if starch was present, the test tube solution would turn a dark blue-black color. If the solution would turn a blue-black color, we would record it as a positive test result, and if there was no color change other than the brown color of the LUGO solution, we would record it as a negative test result. After that, we would perform the sulfate ion test on test tubes 2 and 6. So for this, we would add several drops of the 2% BaCl2 solution to each test tube, and if sulfate ions were present, a white precipitate, known as barium sulfate, would form. So if that would form, we would record it as a positive test result, and if there was no precipitate, we would record it as a negative test result. After that, we would perform a chloride ion test on test tubes 3 and 7. So we would add several drops of silver nitrate to each test tube, and if chloride ions are present, a milky white precipitate, also known as silver chloride, would have formed. And if the precipitate would form, we would record it as a positive result. If there was no precipitate, we would record it as a negative result. And lastly, we would perform the protein test on test tubes 4 and 8. So for this, we would add several drops of the Bure reagent to each test tube, and if protein was present, the solution would turn a light lavender color. So if the solution turned light lavender, we would record this as a positive test result. If there was no color change other than the bright blue color of the Bure reagent itself, we would record this as a negative result. Initially, our experiment was going well, and we were able to add the LUGO solution to test tubes 1 and 5 in order to perform the starch test. However, I misread this part of the procedure, and so I accidentally added LUGO solution to test tubes 1 through 5. So now we were left with test tubes 1 through 5, all containing LUGO solution, and we were left with no cytoplasm test tubes left in order to test for protein, chloride ions, and sulfate ions. Although we'd still had our test tubes 6, 7, and 8 left in order to test for protein, chloride, and sulfate, we had no basis of comparison for them because our cytoplasmic test tubes were already contaminated with LUGO solution. So, although we were able to add the Bure solution, the BaCl2 solution, and silver nitrate to each of these test tubes, they gave us no information regarding the movement of solutes because the LUGO solution already contaminated test tubes 2, 3, and 4, so that really messed up our experiment. So what we ended up doing was looking on with another group's experiment and also researching on our own to find out the solute movements within these solutions. After performing the starch test on test tubes 1 and 5, we came up with test tube 1, which contained the cytoplasm, having a positive result, and test tube 5, which was the extracellular fluid, having a negative result. And our conclusion for this was that starch was not permeable through the membrane because it was only found within the cytoplasm and not the extracellular fluid. Therefore, no movement of starch occurred. And thus, starch is a relatively large molecule with very low permeability. Moving on to the sulfate ion test, our test tube 2 would have tested positive, and our test tube 6 would also have tested positive, based on our research and observation of another group's experiment. And so our conclusion for this test would be that sulfate ions are permeable through the plasma membrane because the movement of ions occurred from within the tube through to the extracellular fluid. Therefore, sulfate ions are small molecules that are very permeable. For our chloride ion test, our test tube 3 would have also tested positive, and so would our test tube 7, which would also have tested positive. And our conclusion for the chloride ion test would be that chloride ions are permeable to the membrane as they moved from within the cytoplasm to the extracellular fluid. Thus, chloride ions are small and very permeable. However, upon looking in our textbook, I found that our textbook disputed these results as they said that chloride ions, which you can see in the picture from the textbook over here, have a very low permeability. And this disagreed with our observations of the chloride ion test that we observed another group complete. Lastly, we would have performed our protein test on test tubes 4 and 8, and test tube 4 would have tested negative, while test tube 8 would have tested positive. And our conclusion for this test is that protein is not permeable to the membrane, as it did not move from the extracellular fluid from the beaker into the tube. Thus, protein has a very low permeability. And in this case, upon our research within our textbook confirmed these results. As we can see in this picture from it, it says that proteins have very low permeability. And this presentation concludes Lab 6, movement of solutes through a semi-permeable membrane.